First start by sharing the screen. So this is the group in Utrecht, and this was the first picture that was taken right after the lockdown. So everybody was very happy that we were able to meet each other again. And well, with this picture, the whole group would like to say hello to Cuba. So we are the Plant Microbe Interactions Group at Utrecht University, and we do a lot of research on the plant microbiome. We strongly believe that the root microbiome is very important in order to keep the plant healthy, and we might also make use of the knowledge that we gain from our research to make our agriculture more sustainable. So what you see here on the slide, and here you see a root, here you see another root, and the blue color that you see is a secondary metabolite called coumarin. And coumarins are secondary metabolites. They are produced by plant roots when the roots are colonized by beneficial microbes. And these coumarins are secreted into the rhizosphere. And because they have selective antimicrobial activity, they are also able to shape the microbial community in the rhizosphere such that microbes become selected that are beneficial for the plant and pathogenic microbes are inhibited. Well, let's first start by introducing the root microbiome a little bit to you. So we all know that plants get free energy from the sun, and they transfer this energy into photosynthesis products by which they can grow and develop and produce all the nice foods that we are all consuming as human beings. But they do not only produce these photosynthates in order to grow themselves, they also secrete a lot of these photosynthates through root exudations into the soil environment. Up to 20% of all the photosynthates become secreted into the rhizosphere. And you would say, well, this is really stupid. Why don't plants invest it in growth for themselves? Well, it appears that they do it in order to feed the microbial community that is living around the root system of all plants. If you think about it, if you take 1 teaspoon of soil, there is 1 billion of microbes in there. So there's a huge amount of microbes that live in the soil. And if you look closely around the root system, you will see that plants select not all the microbes that are in the soil, but only a selected population of microbes become recruited to the root system. And they do that because these microbes have a lot of functions that are beneficial for the plant. For instance, they can help to recycle nutrients in the soil so that they become better available for the plant, so that the plant can grow better. But also many of these microbes have antimicrobial activity so that they can inhibit growth of fungal pathogens that are in the soil, for instance. So there are many services that are provided by the microbial community that is living around the roots, and it appears that plants can select the ones that are really beneficial for them. Well, the community that lives around the roots, we call that the root microbiome. These microbes do not only have a function in the roots environment, they also give services back to the plant, such as provide enhanced nutrients to the plants, but they also increase stress tolerance. And most importantly, they induce the immune system of the plant such that also the above ground plant parts become better protected against all kinds of pathogens and insects. And we call this phenomena induced systemic resistance. And well, there are many beneficial microbes that live on the root system that are able to prime the defense system of the plant, well, enhancing the protection of the plant against pathogens and insects. So what kind of study systems do we use? If we investigate the microbiome, we use the model plant Arabidopsis thaliana. And in order to investigate the microbiome of Arabidopsis, we often make use of a soil from a natural reserve in the Netherlands called the Clipchamp. And here you see students digging the soil from this nature reserve to be used in the lab. And we use this soil because it has a long-standing recording of Arabidopsis occurrence. Here you see a wild Arabidopsis growing in this soil, and year after year there's a large population of Arabidopsis growing in this soil naturally. So most likely the microbial community that is present in this Clipchamp soil is very favorable for Arabidopsis. So that is why we use this soil in all our studies. Well, already mentioned in my first slide that plants select microbes to their root system, And what you then see is that the microbial community very close to the roots is very different from the microbial community that is present in the bulk soil. We call that the rhizosphere effect. And you can nicely study that by taking samples from the soil and isolate the microbial DNA from that soil, and do the same thing from the rhizosphere, so the area around the roots. And then, we isolate the microbial DNA, we sequence the 16SR DNA of those samples, and then you can get a fingerprint profile of the microbial community that is in those samples. And if you do that, you get pictures like this. Here you see an ordination plot of many different samples, and each dot basically represents the microbial community in a certain sample, and it compares it to other samples, and if they are close to each other, the microbial community, consisting of thousands of microbes, are very similar. So these ones are all rhizosphere samples taken from the rhizosphere of Arabidopsis. They cluster together, but they are very different from the samples that are taken from the bulk soil, which nicely indicates that the microbial community around the roots, which is basically selecting the microbes from the bulk soil, is very different from the microbial community from the bulk soil itself. So if we zoom into a certain family, in the family of the Pseudomonas species, this is a family that is really capable of growing in the rhizosphere of many plant species. And here we see an example of hundreds of different Pseudomonas species that were selected from the rhizosphere of model plants like Arabidopsis, but also from crop species. And they were all sequenced, their whole genomes were sequenced. And if you then look at, what kind of functions do they represent, then we see a number of interesting phenomena. So then we see that these Pseudomonas bacteria, they can really produce a lot of plant growth regulators, such as auxin. And auxin is a plant growth regulator, and many Pseudomonas bacteria can also produce these auxins, and this can stimulate growth of the plant. They also produce plant defense elicitors, which stimulates the immune system of the plant, by which they can become better protected against pathogen infection. Well, these Pseudomonas bacteria encode also many nutrient mobilizers, such as, for instance, siderophores. And these are compounds that mobilize iron from the soil, and they can make it available. And then, plants are able to take it up and then also grow better because, the nutrients are mobilized by the microbial community. And another aspect is that many of these Pseudomonas bacteria can also produce antimicrobials. And these are very often active against, for instance, fungal pathogens that live in the soil. And by producing these antimicrobials, these Pseudomonas bacteria that live in the rhizosphere are able to suppress these fungal pathogens and prevent infection of the plant. Well, in order to study this all in detail, we use a model bacterium in the lab, and this bacterium is called Pseudomonas simiae WCS417. This is a model beneficial rhizobacterium. Here it's growing on the plate, as you can see. Here you see it colonizing the root of an Arabidopsis plant, and in yellow you see the YFP labeled WCS417 bacteria. Well, when you do an assay on a plate, you can nicely see that the Arabidopsis plant is growing, but if you grow it in the presence of the Pseudomonas bacteria, you see that the root system is much better developed. There are more lateral roots formed, and also the shoot becomes bigger, up to 2 times more fresh weight, indicating that this root system is of better architecture. Also, the root hairs are longer when they are colonized by Pseudomonas bacteria, indicating that there's more surface area for the plant to take up water, to take up nutrients, and this leads to a plant growth promotion that is typical for these Pseudomonas bacteria. We also have shown that this is a clear auxin effect, because if you take a reporter line of Arabidopsis that responds to auxin by producing this YFP, yellow fluorescent protein, and if you colonize the plant roots with the Pseudomonas bacteria, you see a much stronger YFP signal, indicating that there's a stronger auxin response than in the control situation. And this is responsible for this lateral root formation and this enhanced growth of the root hairs. But this is not the only characteristic that this WCS417 Pseudomonas bacteria can do. It is also already for a long time in our lab demonstrated that this bacterium can induce systemic resistance, or ISR, and this is basically priming of the plant immune system so that the plant becomes better protected against all kinds of pathogens, including this Fusarium oxysporum that is shown on this picture. So on the left-hand side, you see an Arabidopsis plant that is infected by Fusarium fungus, a lot of disease symptoms, as you can see here. But this plant was pre-colonized by Pseudomonas bacteria on the roots, and after infection with Fusarium, the plant develops much less disease symptoms than in the control situation, indicating that this Pseudomonas bacterium is really capable of boosting the plant immune system, leading to enhanced protection. Well, in the past, we have demonstrated that there's one gene in the plant root, very important for the onset of this induced systemic resistance. And that is a gene that we see expressed here in this reporter root of Arabidopsis in response to colonization of the roots by this WCS417 bacterium. And this is a gene called MIP72. And MIP72 is a transcription factor that is only expressed in the roots. And here you nicely see it expressed in response to colonization of the roots by the Pseudomonas bacteria. Well, if you knock out this gene, then we have demonstrated that we do not get systemic immunity anymore in the plants. So it's really required for the induction of systemic resistance. Well, another thing that we have demonstrated is that this MIP72 transcription factor in the roots is also very important for the production of a secondary metabolite that we know as coumarins. And coumarins are secondary metabolites that are produced by the phenylpropanoid pathway. They are fluorescent under UV light, as you can see here. So here you see nicely a root producing these coumarins in response to colonization of the roots by these Pseudomonas bacteria. So they are produced in the roots and they are secreted into the rhizosphere. Here you can see it in an in vitro system. Very bright fluorescent compounds produced in this liquid culture. And these are the coumarins that are produced in response to these Pseudomonas bacteria. But you can also mimic it by growing the plants under very low iron. And here we have a MIP72 mutant. It also does not produce these coumarins anymore. So this MIP72 has a dual function. On the one hand, it is important for the induction of systemic immunity in the leaves, but it is also important for the secretion and the biosynthesis of coumarins into the rhizosphere. And the link will become clearer later. Okay, so now a big question is, of course, how do plants select beneficial microbes to their roots? Here we see a nice picture of a root and bacteria that are colonizing the rhizosphere, and they become selected from the bulk soil. So most likely plant roots secrete certain metabolites that on the one hand attract certain microbes and on the other hand, well, deter or inhibit other microbes so that you get a nice selection of microbes around the root system. Well, there is a lot of research being done on this topic, and it has been shown that there are many secondary metabolites playing an important role there. Also, primary metabolites play a role, like organic acids or amino acids, and also plant hormones. But today I will focus on the coumarins that play a very important role in the selection of certain microbes into the rhizosphere. So to come back on this MIP72 gene that I mentioned before, here again you see this MIP72 gene expressed in response to colonization of the roots by this Pseudomonas bacterium. Here you see nicely the Pseudomonas bacteria growing on the roots. Here you see MIP72 activated in the roots in green. Here you see a control root where it's very lowly expressed and very highly expressed in colonized roots. If you take a section of the root, you also nicely see that the MIP72 gene is expressed in the epidermal cell layer of the root. No expression at all in the control plant. And if you look at the whole root system, you also nicely see that this MIP72 is activated well, in many different parts of the root system. So it's really strongly activated in response to colonization by these Pseudomonas bacteria. Well, MIP72 is a transcription factor, so it regulates other genes. And one of the most important other genes that is regulated by MIP72 is a gene that we call BGLU42. And it encodes a beta-glucosidase 42. And what is known about beta-glucosidases is that they can cleave off glucoside residues from secondary metabolites. And this is often an activity that leads to, well, activation or deactivation of that secondary metabolite when the glucoside residue is cleaved off. Well, we have shown that this BGLU42 activity is also important for activation of ISR, because if we mutate either MIP72, we do not get systemic immunity anymore in the systemic tissues, and the same holds true for BGLU42. When we have a mutant of BGLU42, we also don't get systemic immunity anymore. And I forgot to mention this, but here we see a reporter line for BGLU42 activity, and you see that it is also nicely activated in the same root cells as where MIP72 is activated in response to colonization of the roots by WCS417, as you can see here. Most importantly, if we overexpress this beta-glucosidase, so if beta-glucosidase 42 overexpressors, they show a constitutive enhanced level of resistance against different types of pathogens. So we believe that this leads to a constitutive expression of ISR. So this is an important thing to remember. That was the part about induced resistance. But I mentioned already before that the MIP72 is very important also for the production of this secondary metabolite, the coumarins. Here we see the wild type plant in response to colonization of the roots by WCS417, or in response to iron deprivation, which, you can also induce it in that way. You nicely see that it is produced in a high concentration. What is known is that these coumarins, when they are secreted into the rhizosphere, they mobilize iron and can make it available to the plant roots so that the plant can take up more of the often lowly available iron, and then this can lead to an enhanced growth promotion. Well, you can also nicely see that in the wild type plants, in the exudates, the coumarin levels are very high, because if you take out the plants from the growth medium, then you still have a lot of fluorescent compounds in these wells, and these are the coumarins. If you do the same experiment with the MIP72 mutants, then you don't see any production of coumarins, or very little at least. And if you take out the plants from the growth medium, you also don't see fluorescent compounds in those wells. So from this we conclude basically that MIP72 is a very important transcription factor required for the production of coumarins. Then I come back to this BGLU42 gene that I mentioned before, that is regulated by the MIP72 transcription factor. If you use the BGLU42 mutant and you apply WCS417 Pseudomonas bacteria or iron deprivation, then you can nicely see that they still produce coumarins, as you can see here. In the MIP72, no coumarins at all. In the BGLU42, we still have production of the coumarins. But if you look carefully, they stay inside the roots. Also, if we take out these plants from the growth medium, there are no coumarins left behind in the growth medium, indicating that the coumarins stay inside the roots and they are not secreted in the BGLU42 mutant. Well, from this we conclude that BGLU42 is important for coumarin secretion. Well, then Jannes in the lab, he did detailed metabolite profiling to identify the coumarins in the wild type plant, in the BGLU42 mutant, and in the MIP72 mutant. To make a long story short, he was able to demonstrate that the coumarins that are produced in the roots of the BGLU42 mutant, so the ones that stay inside the roots, is scopolamine to a large extent. Scopolamine is a very well-known coumarin, and it has this glucoside residue, but this one stays inside the roots. In the wild type, we see a lot of scopoletin, which is secreted to the rhizosphere, and that is the same as scopolamine, except without the glucoside residue. And we have demonstrated that the BGLU42 activity, the beta-glucosidase 42 activity, is required for cleaving off this glucoside residue so that you get scopolamine, and scopolamine is then secreted into the rhizosphere. It becomes mobile and is then secreted into the rhizosphere. Another thing that is known about coumarins is that they can have an antimicrobial activity. So then we thought, well, maybe this coumarin is very important for shaping the microbial community in the rhizosphere. And that's something that we tested. First, we wanted to know, are these coumarins affecting the Pseudomonas bacteria that induce them, basically? And that appeared not to be the case. So if you grow Pseudomonas simiae WCS417 in medium with increasing concentrations of scopolamine, then it grows happily. So it is totally insensitive to the coumarin antimicrobial activity. If we then look at some soil-borne pathogens, such as this Fusarium oxysporum, which is a very powerful soil-borne fungal pathogen, it is very sensitive to coumarin because increasing amounts of scopolamine in the medium leads to a strong growth reduction of this pathogenic fungus. And the same holds true for this other fungal pathogen, which is soil-borne, Verticillium daliae, also very sensitive to coumarin activity. Well, then we did a microbiome analysis to see what kind of microbes are sensitive to coumarin antimicrobial activity and which microbes are insensitive. Well, this is again a microbiome experiment in which we looked at the microbial community in the bulk soil, in the unplanted soil. We looked at the microbial community in the rhizosphere of roots that produce coumarins, like in this wild type plant. And we looked at the microbial community in the rhizosphere of plants that do not produce coumarins, such as this coumarin biosynthesis mutant F6H1. Then you nicely see, again, the rhizosphere effect, right? The microbial community in the bulk soil is very different from the microbial community in the rhizosphere of plants growing in that bulk soil. And this is from the wild type. And if you compare it with the microbial community in the rhizosphere of coumarin mutants, then you nicely see that there's a shift in the microbial community. Well, then we did shotgun metagenome sequencing. That means that you just sequence all the microbes in your sample, and then you can identify which ones like to grow in the presence of coumarin and which ones do not like to grow in the presence of coumarin, because we did it on the wild type and the coumarin mutant rhizosphere. Then you see that this selection of microbes, they really do better when plants produce coumarin, and this selection of microbes, they are strongly inhibited when coumarins are secreted into the rhizosphere. Here we summarize what I told you so far. So beneficial bacteria, such as the Pseudomonas bacteria that I talked about, they induce the expression of this transcription factor MIP72 in the roots. This leads to the biosynthesis of coumarins, such as scopolamine and scopolamine. BGLU activity, which is regulated by this transcription factor, is required for the conversion of scopolamine into scopolamine. That leads then to the secretion into the rhizosphere. And there, the scopolamine has a dual function. On the one hand, it mobilizes iron from the soil, which leads to enhanced iron uptake by the plant and growth promotion. And on the other hand, it leads to selective activity on the microbial community. Some microbes tolerate the coumarins, other ones are very sensitive. And then, we tend to believe, but we still need to prove that, is that many of the tolerant microbes, such as the pseudomonads that induce this whole biosynthesis pathway, they are beneficial, and this is the way how plant roots are able to, recruit beneficial microbes to the root system while fending off pathogenic ones. Well, another thing that I already mentioned earlier is that if you overexpress the BGLU42, then you get a constitutive induced systemic resistance, indicating that, the production of, for instance, scopolamine that is mediated by this BGLU42 activity might be important for the onset of induced systemic resistance. So here we see again the system. If you overexpress BGLU42, we get enhanced resistance. This is the wild type, and these are the overexpressors, 3 different lines. If you overexpress BioCubaAgro2022, we get an enhanced resistance against the pathogen Pseudomonas syringae pathovar tomato. Not only against this pathogen, but also against the fungus Botrytis cinerea and against the oomycete Hyalum peronospora arabidopsidis, which is a oomycete pathogen. So then we come to the scheme here. So we have MIP72 as transcription factor, leads to, of course, the biosynthesis of coumarins, and BioCubaAgro2022 activity leads to the deglycosylation of these coumarins, which then leads to the secretion into the rhizosphere that I mentioned before. But now we hypothesize that becoming mobile, leading to secretion into the rhizosphere, might also mean that they are becoming mobile and being transported to the foliar tissues of the plant. And we are now hypothesizing that some of these coumarins that are then produced in the roots travel to the leaves and there induce systemic resistance. And this is an hypothesis that we are currently testing. Well, at least we have demonstrated that these coumarins can indeed travel to the leaves. But before I show you that, I show you first this slide, because we can nicely visualize the coumarins inside the roots. So this was done in the lab of Christian Dubois in Montpellier in France. Here you see in purple scopolamine, in green fraxin, and in yellow esculin, which are three dominant coumarins in Arabidopsis. We also did experiments in response to colonization of the roots by the Pseudomonas bacteria. You nicely see accumulation of fraxin in the roots and also scopolamine in the lateral roots of the plants that are colonized by the Pseudomonas bacteria. Well, we also showed that they do not only accumulate in the roots, like you see here. So here you see the control plants, control roots, and these are roots that are colonized by WCS417. And you see in time a nice increase in scopolamine levels, not only in the roots, also the shoots show enhanced levels of scopolamine. And the same holds true for scopoletin. You should also enhance levels of scopoletin in the roots, but also in the shoots, indicating that these coumarins can travel from the roots to the shoots. And we are now in the process of demonstrating that this is a signal for induced systemic resistance. Well, then I want to switch to another topic because one major question that we already have for a very long time is why and how do these plants recruit immune system inducing microbes to the rhizosphere? How does it work? Well, if you look in literature, then you see many examples that when plants are undergoing certain stress, let's say a phosphate stress, when they grow under very low phosphate, then we all know that the plants start to attract mycorrhizal fungi, which are also part of the root microbiome. And they secrete certain signals like strigolactones. They signal towards the mycorrhizal fungi in the soil, and then you get enhanced colonization of the roots, and the mycorrhizal fungi help the plant to take up more phosphate. So in response to a stress, the plants start to signal, and this leads to help from the microbial community in the soil, and then those microbes help the plant to alleviate this abiotic stress. Well, in the past years, we have also demonstrated that also in response to foliar pathogen infection, in this case downy mildew infection, that plants start to recruit microbes to their root system that in turn induce systemic resistance, leading to enhanced protection against this downy mildew infection. Well, this is something that I'm going to demonstrate in the next slides. So what kind of system do we use for this? So I already mentioned it before. So we use downy mildew as a pathogen. We apply that to the leaves of Arabidopsis plants. And when we do so, we can monitor the microbial community in the rhizosphere. And then we see that in response to leaf infection, the microbial community in the rhizosphere starts to change. And we have demonstrated that there is a group of microbes that become highly abundant on the root system in response to downy mildew infection. And when we isolate those microbes and we provide them back to the plant, we see that these microbes are able, as a consortium, to induce systemic resistance that leads to enhanced protection against this downy mildew pathogen. You can imagine that for this plant it is too late because it is already infected. But you can imagine that, well, if seeds drop from this plant into this soil that was conditioned with these recruited microbes, that is shown on the right-hand side, that is something that we also demonstrated. If you grow plants in this soil and you infect them, then you also condition the microbial community in this soil such that it is enriched for protective microbes. And if you grow then a next generation of plants in that soil, then that plant becomes automatically induced systemic resistant to the downy mildew pathogen. Well, I'll show you the experimental setup that we use for this to demonstrate this. We call that also the soil-borne legacy experiment. So we start with the first generation of plants in pots. On the left-hand side, you see pots with mocked treated plants. They are healthy. And on the right-hand side, you see a pot with plants that are infected with the downy mildew pathogen. We hypothesize that after this downy mildew infection, that the microbial community is changing around the roots and is different from the microbial community that is on the healthy plants. Then we take away those plants and then we grow a next generation of plants in those conditioned soils. And then you have, of course, soil that was conditioned by healthy plants, and you have soils that was conditioned by diseased plants. Then you grow new plants in that soil, and then you inoculate them again with the downy mildew pathogen or not, and then we score for disease symptoms. And then we nicely see that, in this case, we scored for the number of downy mildew spores produced on the plants, that is significantly reduced on plants that grow on a soil that was previously grown with downy mildew infected plants. So the disease is much lower on these plants than the disease on these plants that were grown on soil that was previously conditioned with healthy plants. So apparently the microbial community that is recruited here provides protection in the next generation of plants against the downy mildew infection. Then, of course, the question is, how does this work? Well, first of all, I would like to show you that indeed the microbial community is changing. Here you see a network analysis of all the microbes that are present in the rhizosphere on healthy plants. So these are basically 12 different clusters of microbes that are on the roots of a healthy plant. And then you can see how those microbial communities change in response to pathogen infection. So in green are all the microbes that become more abundant on the root system after downy mildew infection. And in purple, you see all the microbes that become less abundant on the root system after downy mildew infection. So there's really a change in the microbial community in response to infection by the downy mildew pathogen. Well, we were able to isolate certain microbes from the ones that become increasingly abundant. And here you see that on the red bars. These are the ones that over time become really highly increased after downy mildew infection on the root system. And that is a Stenotrophomonas, Xanthomonas, and the Microbacterium. Well, the most important thing is, of course, if you combine these three microbes in a synthetic community, and you provide that to an Arabidopsis plant, and you see whether this synthetic community of three microbes induces resistance against the downy mildew pathogen, you can nicely see that these three microbes induce resistance, leading to an enhanced level of protection against the downy mildew pathogen, which demonstrates that the microbes that become recruited, they are recruited because they have a very good potential to induce resistance against the downy mildew pathogen that started the recruitment. Then the next question is, of course, how does this work? So how do these microbes become recruited to the rhizosphere of infected plants? Well, then, of course, the first thing that we wanted to look at were the metabolites that are secreted into the rhizosphere after infection of the leaves by the downy mildew pathogen. So then Young, she set up a system in which she grew the plants, and then she infected them or not, and they are growing into a hydroponic system where we can collect the root exudates. And if you then analyze the root exudates, then you see that the root exudates of the mock treated plants here in red are very different from the metabolite profiles of the downy mildew pathogen infected plants, as you can see here. Here you see nicely the root exudate metabolite profiles. It's very different in the mock-treated plants in comparison to the downy mildew pathogen infected plants. Then, of course, the question is, the coumarins that I talked about earlier, are they important in this effect? And the answer to that question is yes. Well, how do we know that? We not only because of these metabolite signatures, but also because we were able to do the soil-borne legacy experiment, but now not only with the wild-type plants, but also with the coumarin mutant MIP72. So if you grow your plants in the soil, the wild-type plant, and you inoculate that with the downy mildew pathogen, and then here in red, so in red, the closed symbols are the microbial communities in the rhizosphere of healthy wild-type plants. If you look at the microbial community in the rhizosphere of downy mildew pathogen infected wild-type plants, those are the ones here with the open red symbols, you see a nice shift in the microbial community. So pathogen infection leads to a shift in the microbial community in wild-type plants. But if you do the same experiment with the coumarin mutant, here in blue, and the closed symbols are the healthy plants, and the open symbols are the infected MIP72 plants, there's no significant shift in the microbial community. And again, we see, of course, a very strong rhizosphere effect. So these are the microbial communities in the unplanted bulk soil. And here we see the shift, which is a HPA, downy mildew infected shift. But the blue symbols, we don't see a shift at all, indicating that the coumarins are very important for, well, mediating the shift in the microbial community. Then, of course, a very important experiment is the soil-borne legacy experiment, in which we again try to demonstrate whether plants that are growing in soil that was previously conditioned by diseased plants, then if you take in the first generation diseased wild-type plants, and in the second generation also diseased wild-type plants, we see again this nice inhibition of disease severity in the second generation. So this is basically what I showed you before. So this is the disease severity in the wild-type plants that grow in soil that was previously conditioned by healthy plants. But if you grow the plants in the soil that was previously conditioned by diseased wild-type plants, then you see a strong reduction of the symptoms caused by the downy mildew pathogen. But if you do the same experiment with coumarin mutants in the first generation, from which we know that this does not lead to a shift in the microbial community, and you grow in the next generation wild-type plants on those conditioned soils, you don't see enhanced protection against the downy mildew pathogen, as you can see here. So from this, we conclude the following. So pathogen infection, in this case, downy mildew infection on the leaves, leads to a change in the microbial community. This is dependent on the activity of coumarin biosynthesis, and these coumarins mediate the microbial shift in the root microbiome, and this is very important in order to induce resistance against the downy mildew pathogen in the next generation. Well, I talked a lot about coumarins, but actually they are only the tip of the iceberg that play a role in shaping the root microbial community, because coumarins are not alone. If you look at the roots of plants, there are many structural and chemical barriers that can influence microbiota that grow around the roots system. So I talked about the coumarins, but also other chemical barriers such as lignin or suberin or glucosinolates, or structural barriers like cutin or callose. They influence also the exchange of nutrients between the root and the rhizosphere, and also the secretion of metabolites into the rhizosphere. So we are very much interested in what is the contribution of all these different structural and chemical barriers in the formation and the assembly of the microbial community in the rhizosphere, and how does this relate to enhanced performance of the plant in terms of enhanced growth or enhanced protection against the pathogens. Well, the last part of my talk, I would like to zoom in a little bit on, well, how do we discover which structural or chemical barriers are important in the plant roots in response to colonization of the roots by, for instance, this beneficial WCS417 bacterium. And in order to investigate this, we followed an approach by which we wanted to look at the gene expression in the different cell types of the roots of Arabidopsis in response to colonization by these Pseudomonas bacteria. So for this, we used 5 different reporter lines. So a reporter line that express GFP only in the A trichoblast, which are the cells that form root hairs, only in the trichoblast, these are the purple ones, only in the cortex, these are the blue ones, only in the endodermis, these are the green ones, and only in the vasculature, which are the dark blue ones. So what you can then do is you colonize those roots with the WCS417 or not. After that, you make protoplasts of those roots, and you do cell sorting using a FACS, do RNA-seq, and then you can look at the expression of all the genes in that specific cell type. And then one of the interesting things that we observed was that if you look at the number of genes that are induced in response to colonization of the roots by Pseudomonas bacteria, then we nicely see that not the root cells that are in immediate contact with the bacteria respond the best. Those are the orange and the purple ones. They do respond, but the ones, the cell types in the middle of the root, the cortex and the endodermis, they respond much larger than the cell types that are in immediate contact with the Pseudomonas bacteria. But the cell type inside the root is responding most strongly to the colonization of the roots. As you can see, you can nicely see that if you do an ordination plot of all the different cell types, you nicely see that each cell type responds differently to the colonization of the roots by the Pseudomonas bacterium. And then one interesting finding that I would like to share with you is if you look at these green cells, so these are the endodermis cells that are located here, they respond really strongly to colonization of the roots by WCS417. And if you look at it, then these are all the genes that are responding in the green cells, so in the endodermis cells, and they are significantly enhanced in comparison to the control situation. And there's a nice cluster of genes that encode suberin, which is a compound that is basically a structural barrier in the root system. And we could even demonstrate that indeed responds to colonization of the roots by 417 by using a reporter line GPAT5, which is one of the genes that is induced. And you nicely see that it is indeed induced in the right cell type. Well, here you also nicely see that colonization of the roots by WCS417 leads to stronger suberin staining of the root system, indicating basically that the plant root responds to colonization by producing this enhanced structural barrier. If you use a mutant of this suberin gene, this mutant here, and you colonize it with WCS417 and you compare it to the wild type, you will see that this suberin mutant is much better colonized by the Pseudomonas bacteria than the wild type. So from this we conclude that this suberization is very important for creating a balance between the number of microbes that are colonized on the root system. So with that, I actually would like to end, because in the future we would not only like to investigate the coumarins in more detail, but also all the other chemical and structural barriers that we know are present in the root system of a plant. And we would like to know their function in, well, the communication between the plant root and the root microbiome, and what does this mean for better performance of the plant in terms of nutrient uptake and induction of systemic immunity. Well, this knowledge is very important because in the future we would like to make use of this knowledge to, well, maybe develop better bioinoculants so that we can sustainably protect plants against pathogens and insects, and maybe also use less fertilizers in order to let the plant grow better by making use of its microbial community in the soil. And with that, I would like to stop. Thank you very much for your attention. And this is the group that did most of the work. And on the right-hand side, you see the funding sources that we had to make this all possible. And with that, I would like to stop, and I will stop sharing the screen as well. And then well, I would be happy to take any questions. Okay, thank you very much, and see you around. Bye-bye.